So I will introduce myself. I'm Sonia Villapol. I'm assistant professor in the Department of Neurosurgery at the Center for Neuroregeneration at Houston Methodist Research Institute. And my lab is focused on traumatic brain injury and stroke and Alzheimer's disease, and using different approach. Today I'm going to talk about the microbiome and for diagnostics and treatments of brain injuries. So in the title of my talk, I have I want to focus on one specific project that we have, that is the concussion. So we are looking at the contact sports and different look at the pathology of the different animal models of contact sports, and as well we have a clinical research in athletes. So I think that is a interesting topic because we can focus on the diagnostic, since that there are really bad diagnostic for concussions, and maybe the sequencing in the gut microbiome can be a good approach. And then secondly, as well important, a better treatment and managing of the diagnostic and of this concussion. So as a neuroscientist, we had to start to think about that the brain doesn't act in isolation from the body, that is connected with everything else. And I think that is a major mistake that we did in the past, and we are thinking that everything is on the brain and we don't need to care about the periphery because it doesn't matter. But it's, I think that most of the treatments fail because the reason. So we need to think that the brain is connected with the periphery, and something that happens in the brain can affect different organs, including the gut, and the signals going back. So there are this bidirectional communication that is always relevant, and as a neuroscientist we need to think outside the brain, and that is important to treat the brain, but also to treat the peripheral organs. And related with that, we have the compound that is the brain-gut microbiome, that the human microbiome, as you know, I don't know, you have these numbers that were quite amazing when I read about that. We have 39 trillion of microbial cells that represent 57% oral cells in our body. If we look at the genes, we have 3 million the microbial genes that only that represent 99% of our genes in our own body. So it's quite amazing thinking about the system biology when we don't know that what kind of the functions these genes can have in our own body. So it's important to incorporate these microbiome systems to focus on treatments and understand what happened in the disease. So where the microbiome, these bacteria, they are located, they are in the mouth and the respiratory system and the urogenital tract and the skin, but basically mainly they are located in the gut. So in the intestines, stomach, and there are all these bacteria that live with us in symbiosis, and it's important to get this regulation. So we have some alterations in the system for some reasons that can cause the gut microbiota dysbiosis. So that can affect to different organs and can be the source of different disease, right? So we have infection in cancer, heart disease, Crohn's disease that was close related with the change in the microbiome, asthma, obesity, and as well in the brain. There are several studies that show association in the gut microbiome dysbiosis and Parkinson, Alzheimer's, stroke, autism, and as well in traumatic brain injury. So the traumatic brain injury can cause a neuroinflammation, right? So there are signals that go through the brain, through the periphery, and can interact with the epithelial cells or through the different, the vagus nerve, etc. And this interaction can cause intestinal dysfunction. So there are an increase of the intestinal permeability that induce disbalance, like problems, digestive problems, vomiting, nausea, etc. So this is something quite common when someone gets a concussion. One of the first symptoms is like they throw, like, vomits, and they have really stomach pain and all this disturbance. So there are these evidence that say that the brain injury or concussions can alter the gastrointestinal dysfunction with different symptomatologies. For example, they can increase the intestinal permeability, this thing that called leaky gut, that can be the entrance of pathogens and can cause infections, etc. Can compromise the intestinal mucosa. A concussion can induce the breakdown of the BBP, the blood-brain barrier, so that can induce the neuroinflammatory response, even increments. The neuroinflammatory response can induce brain immune dysfunction, impair gut motility, etc., etc. So there are a lot of affectations of the concussions on the gut. And what happened after, who these things that are mediated. So there are different mechanisms that can communicate the brain and the gut. So we can discuss three of them. For example, the immune and neuroendocrine signaling. There are these cytokines and chemokines and gut hormones that are released through the, after some neuroinflammation in the brain through the gut. There are a direct pathway that is the vagal and spinal afferent neurons that communicate directly to the brain with the peripheral organs, as well with the gut. And as well, this another direction that is the one that starts in the gut with the bacteria and this microbial that can produce metabolites or neuropeptides as GABA, etc., or cytokines, etc., that can alterate the immune system, as well can interact with the neurons in specific areas of the brain. So that is clear that there are a bidirectional communication in all these interventions that we can address across in these pathways that will be relevant. So this is important that we need to understand the gut microbiome dysbiosis after any neuroinflammatory disease, because this bacteria can be alterated, so that can intervene, maybe can intervene the short chain fatty acids or the production of these compounds that are beneficial for the good health. So that is not new, and we know that the epithelia and the intestines, they contain the enteric nervous system, so the enteric neurons. Since now, in the last 10, 20 years, we know more about the microbiome, about these bacteria, but the major communicator between the brain and the gut, there are these enteric glia and enteric nervous system that even 100 years ago, Santiago Ramon y Cajal drew in these beautiful cartoons, and he discovered, and this is an intestine of the gut, and he identified their neurons. So we know that the enteric neuron system has these direct connections, and we have around 500 millions of neurons in the gut. So that is the potential faster pathway that communicates the brain with the gut. So anyway, what happened after microbiome, and what happened after a concussion in the microbiome composition, why we want to know that. So can a concussion impacting our gut head, and why that is important to know the bacterial composition after a concussion. So basically, we need to identify better biomarkers. Most of these concussions during the practice of the sport trainings on games, so there are not identified because there are no apparently biopathology and even neuroimaging analysis. They don't identify this damage, so it's important to have more sensitive biomarkers for concussions, and even as well the blood tests that are not specific enough. So that is clear that the brain injury, the impacts in any sport or traffic accidents, etc., so can impact in the gut microbiome, and that has a consequence because it's essential neuromodulation of the inflammation and the traumatic brain injury outcomes. So we know as well that the microbiome dysbiosis has been linked with several diseases involved in metabolic disease, immune dysfunction, and of course the mental and cognitive impairment. All these players, they have several, they are playing, for example, the football players are playing repetitive, different, during a few years, they can develop an early neurodegenerative disease, as can be Parkinson or Alzheimer's. So we start this study. Before that, I want to, well, let's start here. I move some slides and the other. So we start a collaborative study with Rice University and the neurology department at Houston Methodist. I have an excellent medical resident, neurosurgery resident, Said Sandrameli in the lab, and then our neurologist, Ken Podell, that is the person in charge of the neuropsychologists and neurologists from the football team at Rice University. So we established this collaboration, and we collect samples. We enroll the college football athletes on this clinical study. So we collect samples, saliva, stool samples, blood samples. We perform transcranial Doppler laser on the brain of the athletes after a concussion in every 3 or 4 times during the football season, and the mid-season, that is during the games, and the post-season, and as well when they are resting, that is called the off-season. So we have some demographic data. This data is the players, they are all males. They are more or less average of 20 years old, and they have several parameters that we evaluate depending on the specific clinical demographic for all them, right? So we collect the samples at different time points, and we want to look at we're starting to analyze the transcranial Doppler laser if they have some inflammation in the brain. That was the first setting to measure. We didn't find anything, any blood pressure or even cerebral blood flow alterations using these measurements, even after a concussion. So we want to then analyze the experimental design with the sample collection. We look at the microbiome. So we collaborate with these excellent students from Rice University and doing the bioinformatic analysis. We did in the lab the sequencing analysis using the Minion and the nanopore system, and we did the bioinformatic analysis with different kind of the studies. The longitudinal study, that means we following the same players through different time points during the season, and we analyze the samples 3 time points, and then another extra analysis when they get a severe concussion, a diagnostic concussion. So during the season, they have repeated hits even during the games or when they are training. So we want to compare both, and we found that there are an increase of the diversity of the players when they are during the games if we compare when they are resting in the off-season. So we find this alpha diversity changes. If we want to look at the phylo level, family level, and genus level of this microbiome, on the gut microbiome, we found similar families and the phylo level as well, only when the players they were active, when they were doing the games, if we compare when they are resting. So something was going on when they are, they have different species of the bacteria when they are playing, and then they change a little bit when they are resting. So we thought that we identified that through all the players, like in general. When we do the heat map for the specific bacteria, at the species level, we identify some of them, for example, the Artobacter or the Sphonphilium indicum as well, that there was a stringent difference, was a reduction of this bacteria when they are not playing. What happened with that was through the whole season, but what happened when we want to diagnostic a single concussion? So we got only 4 of the players, they get a severe and diagnostic concussion by the neurologist. So we have a sample that is not so big, but was significant enough to difference to establish some correlation. That was N of 4, so 4 of these players, they get a severe concussion. And so that means that they cannot play, they need to rest for a while. And we found that only in these players there was a difference in the diversity, in the beta diversity of the bacteria. When we look at the phylum level, family level, on the phylum level we found as well only in the players that get the concussion was a difference in the phylum. If we compare with the off-season. And then if we want to create like a profile of the relative abundance of the specific bacteria used after a concussion, a diagnostic concussion, we found some representative families of bacteria, for example, the Landospeciaria, that is from the Clostridia, that is a kind of the bacteria involved in the maintenance of the overall gut function. So it was a decrease after a concussion, so it was alteration of this family of the bacteria. And curiously as well, we found that the one species, like the Bacterium rectale, that is one of the major producers of the SCFA, that is a beneficial compound. So it was a decrease only after in the concussed group. That we identify more species, not only this one, so it's clear that each player we can generate a profile of the species that changed drastically after a concussion, but specific, this one was identifying another disease, as for example in amyloidosis. We found that the other studies show that the Bacterium rectale was decrease in these neuroinflammatory conditions, and these patients is related with peripheral inflammation and cognitive impairment, and as well was identifying neurodevelopment disorders in pediatric, in autism, etc. So, and there are several clinical studies that identify this drastic decrease of the Bacterium rectale as a biomarker for neuroinflammation, even peripheral neuroinflammation or central inflammation. So we want to keep looking if we can identify other biomarkers, not in the stool samples, but maybe in the saliva. We check the oral microbiome. We analyze the oral microbiome through the football season, different time points. We didn't find any significant difference, even in the alpha diversity or phylum, family, genus level, no difference at all the time points. When we look at the concussion group, the concussed players as well, there are no difference at all. We didn't identify any different even in the species level. So probably there are no major changes, and the concussions or the repetitive hits that they have during the season doesn't affect the oral microbiome. And what we want to do next, we know that there are a lot of controversy in the literature regarding to the sensitivity of the biomarkers in blood. So there are some publications that they find a change in the tau levels and blood and the football players. And other articles, they don't show any change on the tau or serum neurofilament. That is the major pro-inflammatory biomarkers in plasma. Another one as well, the GFAP or the S100 beta, were characterized as a kind of little sensitive for the concussion, but it's not clear and it's not obvious that there are good biomarkers. So we did this analysis with our participants on this study. We look at different biomarkers as 100 beta, serum amyloid A that we identify as well in our animal models, GFAP, and neurofilament, etc. And we didn't find the longitudinal study major difference unless we found the GFAP. There are a difference. There are high levels when they are playing, and then when they are resting, is dissipate. There are not this marker in blood. But curiously, after a concussion, we didn't find anything. That is very important when we want to do a diagnostic of the concussion. We need to prove in blood that there are a sensitive biomarker for this diagnostic. These tests were performed like 24 hours after a concussion, or even before, 12 hours after. So even that, there are no evidence that that the concussion can induce this increase in plasma. So that makes more relevant the analysis of the microbiome to have some characterization or some profile. And something that we want to compare, we need to correlate. What we did, we correlate a specific bacteria with the serum biomarkers that we identify. and plasma, and curiously in the players that they have the Eubacterium restale levels lower, they were found that they have more levels of these pro-inflammatory biomarkers, as you can see here in the stars over there. And curiously was matched with the Eubacterium restale again. So probably that is a good target as a biomarker, and not only thinking about the diagnostic, but as well as a probiotic. We can suggest to establish a treatment with this specific bacteria to the players that they get a concussion to prevent that the inflammation or the chronic inflammation can be persistent over time. So definitely we can conclude that this decrease the Eubacterium restale can be correlated with the peripheral inflammation, and as well that the single concussion can induce change in the gut microbiome, and this change, observed in the serum biomarker with the GFAP as well they're correlated with the Eubacterium restale, and we then find more difference after this severe concussion in all the standard neuroimaging analysis or more serum biomarkers that we identify. Even probably the salivary microbiota is not a good reference for this analysis. And with that, just to conclude that, definitely the change in the gut microbiome could be a useful biomarker for a better diagnostic, and as well, we can think about new therapeutic approach for concussions. And I would like to thank to my lab and the more collaborators at Rice University and the Department of Neurosurgery to perform these experiments. Yeah, thank you.
From your point of view, how do you see maybe artificial intelligence and machine learning helping make your work better?
Yeah, that will be another extra tool to analyze all that. We have learning, machine learning. Some of the places they have some kind of the test in analysis and neuroimaging, and the point is correlate all the data together, put all the points together, and as we did the correlation with the blood biomarkers and all these other neuroimaging parameters. So if we can correlate everything and get a better diagnostic, so that will be very beneficial for them. And the point is to identify this chronic inflammation that overall, over the years, they can affect. So that is the thing. There are not good diagnostic, and put all the dots, all the points together will be better even to do a personalized medicine that they need to be diagnostic and treated.
I mean, clearly you're doing a lot of microbiome research. From your point of view, how many kind of biomarkers are out there that could influence how we treat diseases? Is it everywhere, or just in some cases?
Regarding to the microbiome?
Using the microbiome as a biomarker, as you have.
Yeah.
Yeah.
Yeah, and we have the clinical research that we are performing now. That we have Alzheimer's patients that we identify specific bacteria that definitely change. With the long COVID, we have the same. So with the concussions, any disease that can affect the neuroinflammatory system, the gut microbiome is a neuromodulator. It's like the major component, and not only for diagnostic, but as well, to target specific treatments. Has to be everything, correlated with the blood biomarkers, of course. But I think that this has this potential for all the neuroinflammatory disease.
Thank you. That was a very fascinating talk. I have a question. Since you're dealing with human subjects, right? And these are young people playing sports. So when an injury happens, how do you know that it's only the brain that's been injured? Because some of the videos you were showing were, athletes just bumping body on with each other. So how are you associating the fact that it's a concussion that's leading to the microbiome disruption, as opposed to other injuries, right, that could be associated with a collision of people?
Yeah, in the longitudinal study, of course, all the analysis of the gut microbiome was performed, different specific time points. And so, of course, they get hits on the head mainly, but as well and through the whole body that can cause peripheral inflammation and on the arm, and this is systemic inflammation that can affect indirectly to the brain, of course, or the gut. But after a single concussion, it's really a diagnostic concussion when they hit the head and they get unconscious and they need to go through the clinic and they get a neurological test and all these protocols that they have in clinic to diagnose a concussion. So that is the one that we consider as a diagnostic concussion. So we are sure that they have a strong hit on and also the biomarkers that we are using in blood, the neurofilament, and these biomarkers, they are produced by neuronal cells, so they are mainly released through the blood, but mainly they were produced by their neuroinflammation in the brain.
Thank you.
I was wondering if you collected any information about the sleep habit and any changes in the sleep duration, because we know sleep deprivation can result in change in microbiome and many other issues. So I guess if you are in the mid-season and are playing, you may be sleep deprived. If you have concussion, that may cause a sleep deprivation. So it can have basically directional effect.
Yeah, no, that is a great point, and we love to consider that, but we don't have, even another thing that is more challenging. We like to have in the helmets like some kind of biosensor to quantify how many shoot hits they get every single game. All these parameters, yeah, we like to get integrated and evaluate everything together. The same that the nutritional features, the nutrition, the diet. So we have in the team a nutrition, they eat together, they, all these things, but we don't know what they do over the weekend. The diet alters the microbiome as well, not as strongly as other compounds, but there are too many factors, of course. It's a group very well, there are all males. We want to do the same because we know that there are gender differences after brain injury. We know that all these things, we need to do the same study with the soccer woman, the woman team from soccer, so that will be, because they have as well the neuroinflammation, they get less concussions. So there are too many parameters to take into consideration. As much points we have together, we can interpret better the results, but yeah, that is a good point. We love it.
I guess this adds on to that Amir's question. We are from Houston, VA, and we do research in this field too. So our study, we did sleep deprivation. We looked at people who are chronically sleep deprived. We found Suturella and Pseudomonas were low. But the question is gut microbiome. It depends on so many things. Like, so is it ready for the prime time? Can we, for example, sleep, diet, heart conditions, everybody sort of puts out a study that, okay, this is altering the microbiome. And that too is transient. It can alter, and in a few days down the road, it can alter again. So I guess, are we ready for the prime time for gut microbiome, or even close to using that data in making therapeutic decisions or so?
Yeah, so that is right. There are a lot of changes, a lot of modifications that change these parameters. This is a pilot study only with 33 players. We want to extend that, we get 4 of them, they got a concussion. We are sure that, the 4 of them, they are different, but they have this particular thing that there are specific bacteria, but only change after this concussion. And the clinical data, the 4 of them, they were different. So we want to, and this bacteria was associated with another disease that they are, involved in neuroinflammation. So we want to verify that. We want to verify that is real, that is like that is not, something random. And well, we published the paper, but we want to stand that and associate this decrease of this specific bacteria only when there are these severe inflammatory compound that is very fast, and it's a change that occurs, in a few hours after a concussion. And even we identify the sensitive biomarkers with Simoa or another tools and blood. That is the only thing that we have so far, the blood biomarkers that are using the ultrasensitive analysis. So it's another factor to correlate. And of course, like there are too many compounds that can alter the microbiome, but if this thing is constant with a board group of the participants, maybe we have there some key or some elsewhere that what happened. And it's very easy to modify the diet or do this addition of this specific probiotic and just to see if the inflammation decrease or what happened over time. So the probiotic approach is very easy to address. So I think that is one pilot study. So we like to stand and to verify better. Yeah, of course.
Thank you, Sonia. That was our first session. Thank you so much for being here. We're going to do a 15-minute break. Cheers.